data and the brain, and actually we have a more specific topic here, which we are going to be talking about efforts to use data science approaches integrated with clinical approaches to understand and develop better treatment for Alzheimer's disease. My name is Tim Clark. I'm faculty in Public Health Sciences and Data Science, courtesy appointment in Neurology. We have also Carol Manning, who's a professor in neurology and who directs the memory disorders unit here at UVA Health, and Per Sederberg, who's a professor in associate professor in psychology. He directs the cognitive sciences program at the university. The three of us have worked together in the recent period on developing a proposal and plan for an Alzheimer's disease research center at the university, and we're going to talk a little bit about some of the things that make our approach and what we can do here at UVA a bit unique and special. So Dr. Manning is going to start off by talking about the clinical side of Alzheimer's.
Okay, yeah, thank you, Tim. Yeah, so I'm going to, I spend a lot of time with people with Alzheimer's disease, and as Tim said, I'm the director of the Memory Disorders Clinic, so I'm going to tell you today about clinical features of the disease itself. So the first thing to know is that Alzheimer's is the most common form of dementia. There are currently 5.8 million Americans who have the disease. And really what this is showing you is that there is no part of the world that is spared from Alzheimer's disease, and that Alzheimer's disease, the biggest risk factor for Alzheimer's disease is getting old. And as our population is graying, we are going to have more and more people with Alzheimer's disease. It's an epidemic, and we are not prepared to care for it, to deal with this epidemic. So the way we make a diagnosis of Alzheimer's disease has been on clinical features, but that's a provisional diagnosis, and then a final diagnosis is made based on neuropathology from the brain, looking at plaques and tangles. Amyloid, and here you see neurobrain and Alzheimer's brain. Amyloid plaques and neurofibrillary tangles are the neuropathological features of the brain of the disease. And the amyloid hypothesis has been the leading hypothesis in Alzheimer's disease. And Tim, are you going to speak a little bit more about that, or you cut that out? But anyway.
I'm going to say a few words about it, but why don't you say what it is, okay?
So the amyloid hypothesis is based on the idea that we have this aggregation of amyloid in the brain that is the first feature that is necessary for Alzheimer's disease, and it starts the entire cascade. There has been a lot of research, in fact, most of the research on the neuropathology of Alzheimer's disease has been based on the idea that amyloid is the cardinal neuropathological feature. It was, as I said, only based on tissue that we obtained at death. But through PET scan and through cerebrospinal fluid, we can now image amyloid, and that's A beta. You can see the healthy brain up top, and there is much more amyloid aggregation in the Alzheimer's brain on the bottom. We now can also look at tau, which is what those neurofibrillary tangles are made up of. And tau is really used just for research now, but amyloid, looking at amyloid is now more used clinically. So there is a problem with this, and that is that one is we cannot cure Alzheimer's disease, and as many studies as there have been with amyloid clinical trials, there really to date has been almost nothing that has proven successful. But the field has moved such that it used to be that it was a clinical diagnosis. Now, because we can image amyloid, we can look at people who have amyloid in their brain sufficient to be what we would make a diagnosis of Alzheimer's disease with. However, there are people who have amyloid in their brain sufficient for the diagnosis who do not show clinical features of the disease and live out their lives and never show clinical features of the disease. So we are now in this what we're calling preclinical Alzheimer's disease, diagnosing people with a terrible, devastating disease when they never show clinical features of the disease. So what are the clinical features? The clinical features involve cognitive deficits. There has to be memory impairment. When we think of Alzheimer's disease, we think of memory impairment. And then one or more of the following that you see there: aphasia, so language disturbance, sensory disturbance, visual disturbance, disturbance in executive functioning, which is the ability to do abstract planning, to do things that we think of, abilities that we think of as uniquely human. And these cognitive deficits have to cause impairment in social and occupational functioning. The decline is gradual and continues. People start with mild deficits and end up with severe deficits. Now, a problem with these clinical criteria is that they don't involve some important clinical features, which are the behavioral and psychiatric symptoms. These include agitations, hallucinations, delusions, and these behavioral and psychiatric symptoms are important because these are the things that are most disturbing to people who are caring for people with Alzheimer's disease, and these are the features that are most likely to have a person with dementia end up in a nursing home or a memory care unit. So how we diagnose the clinical features and the cognitive change is that we have a battery of neuropsychological tests that measure all of these abilities, and these tests are well validated, they're well normed by age, by education, by occupation, and anybody in the room could take the test, and if you tell me how old you are, how much education you have, what your occupation is, I could pretty well predict how you're going to do. So when we know someone who has a certain education and occupational history, ages, and they perform below what they should across tests or on some of these tests, that's what we would use to make a diagnosis. A problem with that is when we test people typically once a year at most. We may see them on a good day, a bad day. We don't know a lot about what happens in between testings other than what we hear from family. So here you see, the beginning is that people are normal, the end is they are impaired across all areas of cognition, but there's a lot of variability in between, and we don't know much about individual variability in the time between the beginning of the disease and the end of the disease. So there are some things that we know that can change the trajectory of the disease. Cognitive reserve, and cognitive reserve is the idea that people with more education presumably there's a protective factor against getting Alzheimer's disease. Well, it turns out that that's not exactly true, that people with more education get the disease later, but when they get the disease, they tend to decline more rapidly. And the reason for that is likely that people with more education have more cognitive strategies to work around the decline. Once those cognitive strategies stop working, they decline really rapidly. Diet. We know that Mediterranean diet is protective and can slow the rate of decline. That is a diet with olive oil, with fruits and nuts and whole grains. Exercise. There is really pretty good evidence that aerobic exercise is good for slowing the rate of decline and for preventing slowing the onset of the disease. Vascular health is another one. So things like high blood pressure, smoking, history of stroke, history of heart disease, those are all risk factors. They are also risk factors for vascular dementia, and vascular dementia is itself a risk for Alzheimer's disease. And then finally, mood. Depression is a risk factor for Alzheimer's disease, and people with Alzheimer's disease early on often experience depression as well. So what does Alzheimer's disease look like? This is what it looks like, and I should tell you that a couple of things. One is women are at greater risk for Alzheimer's disease than men are, even when we control for the fact that women live longer. And underrepresented minorities are at greater risk for Alzheimer's disease. In particular, we know that African Americans are at greater risk. But a problem is that the huge amount of clinical research that has been done on Alzheimer's disease is largely been in more highly educated Caucasians. So that means that what we know is actually about a pretty slim group of people. So what does Alzheimer's disease look like to me? This is what it looks like, and that is because very often when I am diagnosing or treating someone with Alzheimer's disease, who I'm actually treating is not just the person with disease, but their caregiver. There are, I want to get the number right here, there are 16.1 million Americans who currently are unpaid caregivers. Our entire healthcare system is completely dependent on unpaid caregivers, usually spouses, but sometimes children. So caregivers tend to be older because they're spouses. We know that they have increased morbidity and mortality, which means that the healthcare system we are relying on to provide care for Alzheimer's patients is a frail system. Caregivers experience increased stress, depression, and caregiver burden relative to people their own age who are not caregivers. And we don't really know very much about the relationships between caregivers and patient factors and the impact that caregivers can have on the trajectory of decline in the patient. So in terms of things that we want to know, or that I want to know, and I hope you would want to know too, are what about dementia and underrepresented minorities who actually are at greater risk for the disease? We don't know much about the symptom of variability or the fluctuations within individuals. We know when we diagnose them, we know when we see them, what happens when they're in clinic, but we don't know what happens in between. We don't know, and we need to know more about the impact of caregiving on caregivers, people with dementia, and the trajectory of the illness. I'm going to turn it over to Karen next.
Cool. So I'm going to move around a little. Stuff up here. I'll be over here. I'll try to jump less. And slideshow. Not working. There. Percent from current slide. How's that look? Cool. I'm going to go here. Hopefully that's all right and that camera can get me. If not, then they'll hopefully hear me. So first of all, thanks for having me here. I'm really excited to talk about this. And I think what's clear from what Carol was just saying, we have a lot of questions still to be answered. And what I'm going to come out here, I'm going to describe where my angle is. I'm a computational cognitive neuroscientist. And so what that means is I try to understand the brain in the same way that a physicist wants to understand the universe, which means I don't give up when I just notice that something happens. That's cool. It's always fun when we see some significant effect. But my goal is to understand the underlying mechanisms, these generative processes that give rise to X, Y, or Z. And I care about cognition. And so I've spent the last handful of years trying to develop ways to quantify cognition in a way that we can actually understand what's happening. And what I hope you'll see today is when I'm going to introduce this general approach to you, and what I hope you'll see is that this is something that can be useful in the domain of Alzheimer's and just understanding how the brain works. And hopefully we can gain some insight and from collaborations that we are actively forming and exploring now. So how did I really come to start this set of work? And that is that when you do these tests, and Carol talked about the way that they currently assess cognition and cognitive abilities in patients and cognitively normal individuals, what will happen is that you'll do these tests and then they'll boil it down into a single number. That's this point right here. Sometimes, so obviously you can't diagnose someone based on a single number, so I'll actually give you the number. 14. You got a 14 on that test. What does that mean? What does it actually mean that you got a 14? Well, maybe I'll get you, someone else will take that test. And they got a 22. All right. Well, is 14 different from 22? I have no idea. Because you have to be able to quantify any level of uncertainty in those measurements. And most of the time, the different tasks that you use have no way of quantifying one point or the other and say that there's actually any variability in there. So what they'll do is that they'll run these tests in thousands and thousands of people and then create some sort of diagnostic threshold. Say if you're above this line, then you are doing just fine, and if you're below it, then maybe we need to evaluate you further. And so this is the problem. This is the status quo of cognitive assessments, and here I'm just showing examples. If you think that I'm exaggerating this a little bit, this is data from ADVI, this large-scale Alzheimer's disease initiative, showing two measures that people often use to assess cognition, the CDR sum of boxes, the MMSE, and these are different diagnostic criteria that you can apply to those numbers. And the problem is that we are relying on groups in order to make these diagnoses, and we don't gain any actual, first of all, as I mentioned, you're not quantifying uncertainty, and then lastly, and this is the part where that my work really comes in, is we're gaining very little insight into what actually gave rise to that score. And one of the problems is we are not doing a good enough job of quantifying the heterogeneity in these populations so that we can maybe have individualized individualized treatment and just come at this problem a little bit differently. So what I've done is I've developed this ecosystem. My lab and I have spent a bunch of years doing this. We call it SUPREME. And that's because it's always nice to have some sort of acronym. And this is from Sensing. This is the dawn of a new way of doing these sorts of quantifying cognition. So sensing from understanding to prediction, realized via an experiment and modeling ecosystem. Now, what do I mean by all that? We developed a cross-platform experimental library so that we can code experiments that we could then deploy out to phones and tablets and laptops and things like that. I'm not going to go into detail about that. It's called SMILE because I like acronyms as well. Then we developed a handful of cognitive tasks. So these are very similar to a lot of the tasks that are in use all of the time now, except they've been designed so that we can do what are called mechanistic model-based analyses. And that's where this last bit and where we spent the most time is developing these computational models of the behavioral performance on these tasks. Now I'm going to step you through, focus on these the last two components here, just so you understand exactly what I'm talking about. And when I say model, for example, if you were here for the last panel, and what we can do with it. So these are standard tasks. If you have ever done any sort of cognitive assessment before or taken a psychology class, you might have heard of a handful of these. The basic idea is that these tasks tap into a full array of cognitive processes. And the cool thing is that we can run these tasks, all of them, in under 20 minutes. Which means that if someone has access to a computer or a tablet or a phone, you can assess someone's cognition in 20 minutes here. And what we're able to assess are things like attention, perception, decision-making, working memory, long-term memory, inhibitory control processes, reward learning. This is a lot of what is cool about what our brains can do and what is going to fluctuate as we they fluctuate on moment to moment, day to day, and how much sleep did you get, what's your blood sugar like, how much exercise did you have, but also, do you have dementia? Are you on a trajectory towards mild cognitive impairment and beyond? So what do I mean by a computational generative model? So most statistical models, so these are everyone's doing a model. Whenever you do any sort of statistical test, it's a model. But most models are what are called descriptive models. What that means is that you are describing whether or not you have some difference in these two distributions. So this might be one group, this might be another group, this might be your performance on one part of the task and performance on another part of the task. And you can assess, you can describe, oh, there is a significant difference between those two distributions. What a generative model does is that it takes, well, what are the processes? What are the cognitive processes that actually generated that behavioral data? We can implement that within a system of mathematical equations. And then, that's why you have the gears on the brain here. And so these system of mathematical equations then generates the observed data. And then we have parameters of those equations that actually govern the latent processes, the latent cognitive processes that track that. So I'm going to try to speed up a little here because I've got a lot of stuff. And we want to have time for it because this is only a 3 hour long.
1 hour long. We need time.
Yeah, I'm just kidding. Okay, so I'll give you a quick example, a concrete example of what do I mean by developing this model here. So this is a flanker task. It's a task that measures inhibitory control. Your job is to look at that center arrow and tell me what direction it's going. And this is where the outer arrows, the flankers, are either congruent or incongruent. This is the incongruent case. And so when people do this task, they're really good if it's congruent, and then if it's incongruent, they tend to make these really fast errors, meaning if they respond quickly, they have an error in there, but if they take their time, then their performance goes up to about the same level as you might see in the congruent condition. And this can tell us a lot about how resilient you are towards facing distracting features in your environment, and we have a lot of cool work that shows that this maps onto real-world scenarios. So we could take this and try to model it. Well, we could take a model of decision making. In one case, we might use something like a diffusion decision model, which says that you accumulate evidence towards one choice or the other, in this case, to the left or to the right. But now we need to modulate that through time, because what people are doing here is they're attending more and more to that center arrow, and they're able to ignore those flankers. So we can pop on top of that some attentional spotlight. And we can have mathematical equations that govern all of these, and then fit the equations to the data, and now then we have a model fit. And we can do this within a Bayesian framework and get uncertainty and all of this fun stuff. So let me give you a couple examples, and then we'll probably skip a good bit of what I have at the end because I could talk for a while. So this is what I mean what we're able to gain from this approach. So typically, you might have a single point. So I've shown you, we had a whole bunch of people do these tasks. And then I've sorted them by their scores. And this is what you get. There's not a lot of heterogeneity. Well, you don't know what it means, because each of these subjects are boiled down to individual points. So these are the percentile measures. Well, someone might argue, a lot of times on these cognitive tasks, they have subscales. Well, here, we'll break those scores down based on their which task we were talking about. And so this is ordered in the same way at the top, but now we have this a little bit more variability here. But what we're able to do with the computational model is break that down even further, and now every one of these values that I'm going to show you is actually an assessment of their cognitive processes that went into performing each one of those tasks. And this is what you get. So what this means, what we're able to see here, is that there's a lot of variability here. And people that are performing identically can have different reasons for performing at that level, and that is key. And so now let me zoom in on just a couple points. So here's two people who performed poorly and two people who performed well. We can now assess whether or not they're significantly different from one another because we can quantify uncertainty with this framework. So now you can see these are the two poor performing individuals, these are the two high performing individuals, and this is specifically, I think, on the flanker task. And so what you can see here is these two are not significantly different. These are distributions turned on their side, violin plots, and you can see that these are overlapping and these are overlapping. However, when we look at their individual parameter estimates, these are the two poor performing individuals, they did just, even though they performed quote unquote identically, the reason that they performed that way were completely different. So when we have parameters that don't overlap here, this means that their actual cognitive process that generated that was different. And so one person was faster, their motor response was faster, but then they also were distracted more by the flankers. So that means their brain's actually doing something different, even though nominally their performance was the same. So we could do this sort of approach now. I've got a validation where we've run this in a large data set in collaboration with one of the Alzheimer's disease research centers. This one is in Indiana, where we took a bunch of cognitive continuous recognition data and from people from controls all the way up to AD. 5 minutes? Yes! I thought I had negative 5 minutes. All right, darn. All right, so the basic idea here is that, sure, people's memory performance gets worse, and they start to slow down as they get older. Great. If you use the summary statistic data to try to predict their diagnosis, you can only account for 7%. Now we can develop a computational model. I'm not going to go into the details of this model, but basically I'm a memory theorist, and we have a lot of ideas of how the brain actually forms and retrieves episodic memories. And we can put this into a model that has parameters that map onto actual cognitive processes. And look, our model can capture the behavior really well. So on the left here, this is the behavior. On the right, this is the model-generated behavior, and those look pretty similar. So what can we do with that? Well, we can assess variability. So you can see that the control was doing well, the early MCI was doing less well, and these two late MCI were doing basically identically here. But the reason that they were performing identically is different. So if you look at all of these light blue comparisons here, their parameters are actually not overlapping in a lot of cases, which meant that one person that is late MCI, it is manifesting. The way that it is affecting their brain is different from the other person. And that's important for how you might treat them. And so what else could we do? We could take those parameters and predict diagnosis, and now we can account for 30% of the variance. So we went from 7 up to 30 by taking the same data. Those data only took 5 minutes to collect, by the way. And the same data, and we account for much more of the variance. That means this is a useful way of characterizing their performance. So I'll skip through here. That's a little summary. I'm going to go to this last little bit here. We don't have to stick to only behavior. We are working to develop what are called joint models that link the behavioral parameters and neural parameters, which means that when we, instead of doing the standard approach to cognitive neuroscience, where you might have a manipulation of your experiment and you look for the consequence of that manipulation on the brain, here we're actually doing what I like to refer to as taking the theory out of the discussion section, because what you'll do in cognitive neuroscience is you'll see an effect and then you'll talk about it all in your discussion section. Here, we bring it right up into the results. And what that means is that we can link together variability in actual latent cognitive processes with variability in neural data, whether or not that's structural data or functional data. So we gain even further insight into the neural correlates of this decline. And so I'll finish there. We can do things like take it out of the lab and bring it into the real world. And the basic idea here is that I want to use this system to now do things like help identify MCI earlier, and also to track fluctuations in cognition, both for, and as Carol was saying, both for the patients, but also their caregivers. So we can learn the relationships between when you have those good days and those bad days. How does it affect the way that your thought processes work? How does it affect your overall cognition? And that's going to be different for every individual, and the interventions that we might do would be completely different depending on how this manifests in each individual. All right, thanks.
So these were both pretty interesting discussion or presentations already, and I'm going to try to tie them together. And unfortunately, or fortunately, this is just a fire hose of information, right? So I'm going to talk about our notion of how to do an Alzheimer's research center. It's very competitive to get National Institute on Aging funding for this, extremely competitive. And there are only 30 centers in the US, and pretty much the same groups have had the centers for ages and ages and ages, right? But they've introduced a program that will give you an exploratory center, and there are 2 possible slots to get these centers. We're going up against a lot of people, but they're looking for very innovative approaches to try to change the mix of people who have these centers, right? So I'm going to tell you a little bit about the heterogeneity problem in Alzheimer's, which is a big problem, and also how we're approaching this, right? So this is Alois Alzheimer and his first patient, right? She was demented, and this was I think 1906. She was demented. How old is she?
1973.
51. Right? These are his brain sections that turned up at the University of Munich in about 10 years ago. And they survived 2 world wars and saturation bomb. Okay? This is how great the Germans are at science and preserving specimens. And one of the interesting things, his second patient was, so what he identified from her brain slices was this whole phenomenon of amyloid plaques and neurofibrillary tangles that Carol talked about. And she got Alzheimer's at age 51. Second patient had Alzheimer's as well. He had very extensive Alzheimer's history in his family, as has been shown by subsequent analysis. And in fact, they were able to take sections, some of these brain sections, and do some genomic analysis on them recently. And they found that the second guy had a presenilin-1 mutation, which is a hallmark of what is called familial Alzheimer's disease, right? It turns out familial Alzheimer's disease really accounts for, I think, just 5% of the cases. It is pretty well understood, but it is not the big problem. The big problem is what is called sporadic Alzheimer's disease, which is 95%. So heterogeneity in cognition, also you have heterogeneity in the types of this disease, and in fact, I'll point to something else. From 2002 to 2017, in AD clinical trials, there's a 0.15% success rate. All right. The failure rate. There's one approved drug, memantine, which is not a very good drug, right? And it only ameliorates the cognition. It's sort of like if you're on the trajectory of decline and you start taking memantine, you come back up a little bit, and you go.
It just slows the rate of decline.
Right. Right, but it doesn't cure the disease, right? So for actual disease-modifying agents, out of hundreds of clinical trials, it costs tens and hundreds of millions of dollars, maybe billions of dollars to run. 100% failure rate for disease-modifying agents. That's if you took this 0.15%, you would say, compared to the generic CNS trials, that's 2 orders of magnitude worse than performance in CNS trials in general. Why is there such a high failure rate? So there's 4 hypotheses. People have sort of gotten saturated with this failure rate, and people in the field are thinking, okay, what are the hypotheses that could explain this high failure rate? Number one, we're treating them too late. So by the time you start to get symptoms, the big lag between the pathological processes in the brain and the development of symptoms. So by the time that you have symptoms, you've lost all these neurons, can't help you, right? Number two, wrong target. You're looking at the wrong molecular target. This is the amyloid cascade theory that Carol mentioned, that amyloid is the agent, is the sort of idiopathological first principle of Alzheimer's, and maybe that's not true. Or 3, AD heterogeneity. There's different processes that are leading to Alzheimer's, and they're all sort of being lumped together as one disorder, where it's really a lot of different disorders. So this is really actually sort of the same thing as number 2, right? Obviously, if you're treating the wrong target of one disease, it's also similar to, well, you might have a slightly different disorder, right? And we're treating the wrong target. So, this is Rhoda Al, who's at the BU Alzheimer's Center, and the quote from her is, if we shifted the presumption to multiple diseases, it changes the whole approach on how you would deal with drug development. So, as I said, we are developing here an Alzheimer's disease research center with a lot of support from the Brain Institute and, I might add, from the School of Data Science. And Carol is the director of the center. Jaggi Kapoor, who also heads the Brain Institute, is the co-director. And we are trying to build an Alzheimer's disease research research program that has pervasive data science approach. Now, when NIA developed the program to have Alzheimer's disease research centers, early on they recognized, well, we need to collect data and let's do some statistics on them. So we're going to require you in your proposal to have a core in your center that has some data management and some statisticians in it. Well, that sounds like a good idea, right? But when they developed this model, there wasn't really a concept of data science, right? This is before there were mobile phones. This is before the World Wide Web really took off. We're trying to update this model in our proposal and in our approach, and really make data science a core part of the whole approach. So there's really two things that I think make our approach unique. One is a clinical core, which is really focused on both the dementia patients and the caregivers. Actually, the cohorts are healthy aging, mild cognitive impairment,
people with dementia, and caregivers.
Right. And we're trying to look at them in an integrated sort of systems way. We're also trying to do incorporate various kinds of innovative testing and all the rest of it, as Eric has discussed. And the other is data science broadly conceived as a core discipline. So the proposal here, we were asked to put up 3 cores, and initially it's an admin core, a clinical core, and a data science core, but developing into adding neuroimaging, adding neuropathology, adding an outreach core to go out in Southwest Virginia and up into INOVA cohorts up in DC and so forth. And over time, data science actually becomes, in our approach, it sort of becomes an integrative factor even for disciplines that are really important to understanding Alzheimer's but are outside the funded cores, like genomics or basic biology and so forth, or biomedical engineering for that matter. So, how many people have read this paper? A few people, right? A few people, but it's actually one of the most highly cited papers in science right now. And it's about making data available, findable, accessible, interoperable, and reusable. And this is a really important model. This is an important concept for the School of Data Science. It's also an important concept in our center because it affects how you can actually share data and make it available, and make how you develop the data, as Vicki Stodden talked about this morning, becomes very important in actually accelerating science, right? So this is from a paper from Baron Mond, who talks about levels of FAIR. This is what he calls FAIR data restricted access. So this is what we're going to be providing, is data whose descriptive metadata where you can find it, but you're not necessarily going to be able to access it because it's controlled, it's HIPAA controlled. We'll try to anonymize a lot of this data, as was discussed earlier, but fundamentally some of this data is going to remain confidential, but you can still go and see where it is and go and apply to access it. So this is kind of the diagram that we put together for our proposal, and I'll just say this is actually kind of a basic data capture environment using REDCap, where you database all your patients and so forth. But once what you really want to be able to do is not only capture data periodically, you're testing these patients and so forth, but you need to be able to extract data from these databases and analyze it using different tools and methods. So what are the tools and methods? We want to have all the project teams that are analyzing this data using as many different appropriate data science methods as possible to help tease out the heterogeneity that is really inherent in this disorder. And part of this is also we're developing a Virginia Dementia Data Commons environment, which is will be integrated with iThrive. And this is kind of the diagram, complicated diagram of sort of the microservices. This is actually, a lot of this is actually implemented already, and we're using it we're trialing it out on time series data from the NICU, working with Doug Lake and Randall Moorman and other folks and Amanda, so to do this analysis. And the way this kind of works, this is what is this? Anybody want to say what it is? It's a heat map, right? What is it? You know what it is, but don't tell me. Okay, well that's the point, is what the heck is this? This is an artifact out of a computational study. You want to know who made this, can I reuse it, where's the data, can I reuse the data, where's the software, can I reuse the software, how can I interpret this, what was the experiment, is the computation fit for purpose, can I adapt this experiment? That's fair data and fair software, right? That's what we're focusing on, fair data and fair software. And this is just a clip from a Jupyter notebook for the analysis that was run. And this is actually using these microservices that you saw in the diagram, which find every object in here gets an identifier, persistent identifier. They could be DOIs. We're using ARCs right now, but it could be DOIs, so it's citable and publishable. When you resolve the ARC, you can return the metadata. It's formal terms from formal ontologies. You also get an evidence graph for every object, which is a directed acyclic graph of the provenance, going back to the primary data through where you did the computation, what computer it was on, who did it, what was the software that was used. Here's the input data set, computed using this software. The software has to be registered with the system and get an identifier before you can run the process, and you can get the software. So all of this is resolvable, right? And it sounds complicated, but it actually makes it easier to do the analysis. So lastly, what I'm going to say is we want to encourage the UVA data science community to engage with the Alzheimer's research that we're doing and developing Alzheimer's Center, and we really want to reach out to people and see if you have thoughts and ideas and potential projects that you'd like to do with us. So that's my little contribution to this, and what I'm going to do is just put the question to the audience. Actually, is first of all, how could the data science community at UVA apply resources or do interesting projects that could help this effort?
Well, I have a question before that.
Okay.
If that's okay. It seemed like, and I might have been misunderstanding, that there's an assumption that a drug is going to be the result of some research finding, but I may have misread that. Okay.
So I think it depends on who you ask, but my assumption is we have been involved in clinical trials for Alzheimer's disease since the very first one, and we have not seen any success.
Right. So that's why I was wondering, like, why. I guess it was that quote from the Boston person referring to a drug that I was like, well, it seems like all the evidence points to that's not working. So wouldn't data need to go like sort of further back to sort of, I don't know.
So the drugs that we have change the rate of decline. They do nothing to briefly for a 3-year period, and then people decline to where they were. We have nothing that actually is disease modifying. And frankly, we have spent a huge amount of money on the amyloid cascade hypothesis that it's not going anywhere.
So the question, so maybe I would say there's I have two answers to that. One is that Carol and our center's focus on caregivers is really, really important, right? And that working with the caregivers can potentially affect the rate of decline, right?
Can change the rate of
environment that people are living with. Right.
So to answer that a little more directly from my perspective, ultimately we want a cure, we need a cure. We're not close. And we have all of these millions of people with the disease that we have to figure out how to help them today, because they're not going to be alive for the cure.
So I have like a follow-up question. Are you seeing that the data you would be collecting would be data that sort of like some of the things that you talked about that you already know, or would you be sort of taking a step back and wanting to collect data that you have no idea if there's any impact on the disease because you haven't gotten anywhere with the amyloid? I guess I'm wondering if the data collection like goes to like, well, this isn't working, so let's collect a lot more data, and then how does?
So I think that it gets back to the topics that I discussed a little bit there too.
Yeah.
There's a lot of data coming in. Data, it's on its own, does you no good. Understanding why those data emerge, so I would say, describing that something exists only gets you so far, and you have to understand what gives rise to these different things. And in this specific case, when we're talking about Alzheimer's, we have been greatly limiting our ability to assess the heterogeneity in these populations. When you limit the heterogeneity, when you are essentially, to use a Bayesian term, when you're giving rise in a hierarchical model term, if you're getting shrinkage and therefore reducing the variability in the way that you assess the population, that also reduces your approaches. And you think, oh, let's throw a bunch of drugs at this and maybe it'll work because everyone is the same. But the problem is not everyone is the same. And so at the same time that we will be, yes, taking lots of data, then we can use these data-driven approaches to maybe get whole new types of classifications of mild cognitive impairment and the trajectories towards this.
Type A, type B, type C.
All
right?
of these different characterizations.
There might be different actual pathological
That's right.
processes in the brain.
Then it's not just drug interventions. There could be lots of different behavioral interventions. There could be lots of different, maybe there's some device you can put on your head.
Right? You need to go run more, take walks.
That's right.
That's your big thing, right?
So what we've been talking about in addition to that is we have a lot of data, but we don't have the right kind of data.
Mm-hmm.
And so we need to change the kind of data we are getting. The treatment we have now is the caregivers. And so what we need to do is understand the relationship between the patients and the caregivers better so that we can use the caregivers more adeptly to help the patients.
Doug.
Yeah, so I'm really having a hard time wrapping my mind around a success rate of 1 out of 413, when if you just did placebos, you would get 5%. I just don't understand how that could happen unless there is some extra threshold to be considered successful. I mean, it just statistically.
Why would it not?
That result there takes into account the placebos because they have every single one of those studies has a placebo condition.
Right, absolutely.
A control condition.
There are double-blind trials with a placebo condition, so there always has to be.
Right.
And there are currently 4 drugs that have FDA approval for treatment of Alzheimer's disease. For symptomatic, none of them change. They have a brief period during which they can slow down the rate of the decline. Patients do not experience improvement by and large, and after a 3-year period, they decline to where they would have been anyway.
So one of the other things I could tell you, having worked in a drug company before, is drug companies are lemmings, right? They are lemmings. First of all, we mentioned the Alzheimer's cascade theory, which is just like scientists can be lemmings too, right? It's like, oh, everybody accepts that this is true, right? Well, maybe it's not. But the other thing is that explicitly in the approval process, to get a drug in your company to get the project advanced to like, we're going to spend the money to take this into humans, right? You have to pass a lot of review panels and a lot of gates. And one of the gates, it's really true, companies will go, was there any other company working on this? And if they are, they go, yeah, well it must be good to work on, right? So they're lemmings.
As you're all well aware, there are a lot of large-scale Alzheimer's disease collaborative networks and consortia where genomic data is generated.
Right.
Even genomic together with image data.
Right.
And behavioral phenotype data. I'm wondering to what extent some of the measures you're collecting, the data you're collecting, can kind of be associated with that data to see if you can infer some of the genetic effects.
Phenotype studies. Right.
Yeah, and kind of leveraging the publicly available data.
And just to be clear, we're already contributing data, the old standard data, to the national databases.
Okay.
And so then the idea here is that, well, maybe some of these new metrics that we're going to be adding into the populations here, we can, and we're already collecting all of the data that would go into these national databases. Well, now if we can build models that link these pieces together, then they can be used more globally. If they show promise, then we can convince the entire networks to incorporate these approaches, and then we're off, and then we've got a lot of data.
And given that you just showed those boxes that were dotted, are you hoping to ultimately get to a point? I mean, I guess you've got to get your grant funded, but hoping to get to a point of generating genomic data and other, like blood.
Well, we'll take blood from all the subjects. We'll take blood from all the subjects. Maybe we'll do lumbar punctures.
And the standard neuroimaging, all the standard stuff.
I mean, Jack, you've been involved a lot in this research. You just came from one of the top centers for Alzheimer's neuroimaging. Do you have any comments about this?
I mean, it sounds like you guys have been also involved in this, sharing data with the Alzheimer's Neuroimaging Initiative, which is the kind of the big multi-center effort that all the data kind of flows to my former institution at USC. And that data, just for anybody who's interested, is openly available. You have to do a little application process, but then you can get insight into all the neuroimaging, all the blood biomarkers and whatnot. I guess my question is what's being proposed here to be sufficiently different than what other ADRCs are up to and what other ADNI sites have been up to? And I think we're into ADNI 3 now. And I think your point is well taken. Everybody's followed this particular model. And just collecting more and more data on this endless, maybe dead-end path, maybe is not sufficient anymore. Maybe we need to be thinking differently. And is what you've embedded in your proposal sufficiently different that it really is a new direction? Do you feel that that's true?
Well, I think
I'd like to think so.
We want to think so. We had discussions with the program manager, and we'll see what the review panel says. But I think in terms of, we're going to be going after other funding other than NIA funding to try to get the center off the ground so that we have other shots at this if we don't get it the first time. But I think, first of all, focus on caregivers is a new thing.
Right. None of the current programs actually spend any time on this.
Right.
That's huge.
And I see that there is a movement, like if you look at the Stanford Center, for example, they're definitely moving towards a data sciencey type approach. They definitely are, right? They have a pretty impressive deep learning expert that has probably 2% time on there, but he's associated with their data management core, right? But actually, I think we're sort of going all in on the data science side, right? And that's, so we've gone to the end of the spectrum on that, and the caregivers thing is new.
So, and I think we had to cover our bases, so we are doing all the traditional stuff and contributing to NAC and everything else, but we also have this new approach that we are added to it, which is really what we think is promising.
So if you look at this as data gathering, but you also look at it as community engagement, is your model going out into the community or asking the community what they want from you instead of saying this is what we have? No, we want to know what you want and then develop something for that.
So that is an excellent question, and part of it that there wasn't time to talk about is we were only allowed to submit 3 cores, but we also have these planning cores, and one of them is called an community engagement planning core, such that that's exactly that. People from the community, where the goal is to reach out and find out how can we engage with the community, how can we both provide them what would be of interest to them and get them engaged in what we're doing.
And I think from my side, so I was associated with the Massachusetts Alzheimer's Center for a long time, and one of the big things that has happened in the community was like education. Really, people wanted a lot of education.
And we already provide education, and my research is with caregivers, and particularly with people of color, underrepresented minorities, so already have inroads into our local community trying to get what they want.
Yeah, so given that drugs aren't working and kind of by the time you're symptomatic, it's too late, what I kind of heard is our best bet are all of these lifestyle choices around what we can do towards prevention. That data tends to be very longitudinal.
Right.
Do you guys have any thoughts on how we could actually capture non-discrete versions of that longitudinal data in a quantitative way rather than qualitative?
Yeah, so that's the goal of the, at least my part in this project. So I'm wanting to quantify cognition. I'm wanting to make it so that, well, it's not feasible for us to have, there are these large scale programs to try to get neuroimaging and from individuals longitudinally, but this is only, it's not feasible to get hundreds of thousands of participants to come in and do neuroimaging with you multiple times. It's just, it costs $500 a pop and then or more, and it's just too much of a time, too much money, and it's just not feasible for us to even analyze all of this data. But 20 minutes of a cognitive task where we have a next generation of what telehealth is. So it used to be that you call someone on the phone. Now we can pump this out. This is the 21st century. Everyone has many people have devices. If you can do cognitive assessments remotely, and it only takes a few minutes a day, suddenly we now have...
We do several months of everyday assessment.
And the idea is now we can change the way that we assess someone's cognition, that maybe you're great on one day and poor on another. Now we're looking at the variability in your cognitive performance through time, and it's a new way of thinking about longitudinal data.
And this may be what you're going to ask, but those lifestyle factors there is good data showing that in midlife, those are things we all should be doing. So things like waist circumference in midlife is predictive of whether or not you're going to get dementia. So what?
My only quick follow-up would be, so in the role of telehealth and stuff like that, where we could have how many times per day the person picked up their cell phone as a proxy for sleep, are we thinking about bringing some of these other lifestyle factors inside of that causal box for exploring in our data?
Absolutely. So one of the other bits of my work is I do, or do you want to?
I'm just laughing about how many times a day you pick up your cell phone. The people I deal with, one, they can't use cell phones, and two, they lose them. They don't even know where they are. So you just revealed your age there. You're doing great. Just keep it up.
But so one of the things that I'm really excited about too is that obviously it's an active, if you're doing a task, it's an active measure, but I want all of these passive data sets. I want access to your Fitbit data. I want your sleep quality. I want what your actigraphy data, heart rate data, all of these other passively collected measures that we can now link up via joint models to your cognitive performance, look at these over multiple time scales, make predictive models of what your life is like and where your trajectory might be, and relate that to your cognitive abilities, then that's a new world. So I just have to collect a few million participants.
So let me also say one other quick thing, and that is, people shouldn't get the impression, because I showed that data and said all the drug trials failed, that we should give up, right? It's just that you shouldn't be bashing your head against that wall over there when there's a door over there. You want to look around and see if there's some doors. And one of the doors also is this whole thing about the lag between neuropathology and symptomatology, right? Can we find some things that are early predictors, right? That are not brain imaging? Because how do you get your PCP to say, well, you're 35, time for going for brain imaging to see if you got amyloid? No, that'll never be funded, right? But how about some things of what you do that could potentially predict this, or combined with the EHR data and genetics, right? So next question, last question, one more. Yeah.
So, I understand about the data part, but don't you also need advances in the pathology hypothesis and all that? If the amyloid hypothesis,
yes,
so how is the data going to drive, I guess, that discovering in the new pathology? I'm just curious if there is a path there or an idea.
Okay.
So there are a couple things. One is, honestly, my interest is someone else has to do that. I am not a basic scientist, but I am the person who deals with the people who are living with the disease right now. And so my goal is to change what I can in the people who currently have the disease. But that said, the data that we are collecting goes to basic scientists who can work on that.
So our advisory panel for this center includes, say, George Bloom, who works in this field.
Right.
It includes Steve Rich, right? So I think, you know, you get the genomics and the basic science, and I think there's an interchange between the clinic, the basic science and the data science, and the community and caregivers. I think that's what we're shooting for.
If I can just
Yeah.
add, there's a lot of fancy mouse models being generated in AD, including sporadic AD.
Really?
Yeah.
So no more familiar FAD?
Yeah. And they have
Really?
the nasty one, but they're generating sporadic ones with humanized amyloid. And, yeah.
But the problem with those models, honestly, is they have amyloid, but they do not have the phenotype of the disease. And so we keep, I mean, for I don't know how many years we've come up against animal models where it's great, there's amyloid, if you don't have the phenotype of the disease, then we're back to the idea that Alzheimer's disease is amyloid, and never mind. I didn't even mention the fact that we see all these people who, for all the world, have the phenotype of Alzheimer's disease, but they don't have amyloid, and then we're stuck with, okay, what is it you have?
Right.
Well, actually, I'll say that the second patient of Alice Alzheimer had amyloid plaques but no tangles. So there's another one, heterogeneity, right?
You also get these super normal people who age quite well into their 90s. They're cognitively with it, sharp as a tack, and you know, look at their brains after they've passed away, and they've got it all, right? They
Exactly.
ordinarily would look like an Alzheimer's disease patient.
So what's going on there? What's the resilience
Yeah,
factor?
we don't have a clue.
So the people think that data science can contribute to a solution here?
Yes.
All right, thank you.